Then we move to specification number 12, which asks us to investigate and state the environmental conditions that affect the germination of seeds, limited to suitable temperature, water, and oxygen. Once again, this topic includes a practical investigation. In this investigation, we aim to find out which environmental conditions are necessary for seeds to germinate successfully. By observing seeds under different conditions, we can determine how water, oxygen, and temperature influence the process of germination and the early growth of a seedling. Before moving into the investigation, it is important to understand the main factors that affect the germination of seeds. Germination is the process in which a seed begins to grow and develop into a new plant, and certain environmental conditions are required for this process to occur successfully. There are three main factors necessary for germination. The first is water. Water allows the seed to absorb moisture and swell, which causes the seed coat, called the tester, to split open. This makes it possible for the embryo plant to emerge from the seed. Water also activates enzymes inside the embryo, increasing metabolic activity so that growth can begin. The second factor is oxygen. Oxygen is needed for respiration, a process through which energy is released from stored food in the seed. This energy is essential for the embryo to grow and develop during germination. The third factor is warmth or suitable temperature. Germination generally increases as temperature rises up to an optimum level because the chemical reactions involved in growth are controlled by enzymes. When temperatures are too low, these enzymes cannot work efficiently, so germination slows down or stops. And carbon dioxide is not required for germination. However, it also does not prevent germination from occurring, so its presence or absence does not make a difference to the process. First, let us discuss the importance of oxygen in germination. In some seeds, the seed coat does not easily allow oxygen to pass through, so the earliest stages of germination may occur anaerobically. However, once the seed coat softens, splits, or becomes more permeable, oxygen is able to enter the seed. Oxygen is necessary for aerobic respiration, which releases the energy required to activate the stored food reserves and to produce new cytoplasm and cell walls for the developing seedling. Next, we consider the importance of water in germination. When seeds are first dispersed, they contain very little water and remain in a dormant state with a very slow metabolism. This dormancy helps prevent the food reserves from being used too quickly and protects the seed from unfavorable environmental conditions. Before germination can begin, the seed must absorb water. Water usually enters the seed through a small opening in the seed coat called the micropyle and then spreads through the rest of the seed coat. Water plays several important roles in germination. It activates enzymes within the seed that start the metabolic processes needed for growth. It also enables stored starch to be converted into sugars and proteins into amino acids, which are used as nutrients by the growing embryo. Water helps transport dissolved sugars from the cotyledons to the regions where growth occurs. It also allows vacuoles and new cells to expand, which supports the growth of the root and shoot. In addition, water maintains cell turgor, keeping the shoot upright and the leaves expanded. Once the plumule and leaves emerge above the soil, water is also needed for photosynthesis. It helps transport mineral ions from the soil into the seedling, and after the radicle emerges, additional water is absorbed from the soil through the root hairs. Another important factor is suitable temperature. An increase in temperature generally speeds up most chemical reactions in living cells, including those involved in germination. Because of this, seeds usually germinate more quickly at warmer temperatures, up to about 40 degrees Celsius. If the temperature rises above about 45 degrees Celsius, the enzymes involved in these reactions may become denatured, which can stop the process and may even kill the seedling. At very low temperatures, around 0 to 4 degrees Celsius, germination may not begin at all in some plant species. However, the exact temperature range suitable for germination varies among different types of plants. Together, these factors ensure that germination occurs only when environmental conditions are suitable for the young seedling to grow and survive.